Hello, everyone. Welcome to Psychedelic Insights. I have a very exciting episode for you guys today. We'll be interviewing the CEO of Cybin, Doug Drysdale. I'm joined by my colleague, Jake Slomowitz. And yeah, we're just going to get the interview kicking off without further ado. Let's get going.
All right. Thank you for coming on, Doug. So we'll start off. So Cybin's phase 1/2A trial with CYB003. Just would like to get your thoughts around the two dosing sessions, 3 weeks apart, and where the idea behind that came from, and how many therapists will be used for this trial for each patient, and also just your thoughts around and the reasoning behind Cybin deciding to let patients remain on SSRIs and SNRIs for this trial.
All right. Well, thank you for having me on, guys. It's good to see you both again. Yeah, look, the CYB3 phase 1/2 study is going really well. It's recruiting well. We're in our second patient cohort, so that's cohort 5, and on track to have efficacy data coming from that study around the end of the third quarter, maybe early fourth quarter this year, but everything's on track. The study design is really based upon evidence. It's based on what we know, all right, to a large extent. So when you look at some of the most impressive psilocybin studies, Johns Hopkins, NYU, and other places where we're all hoping to repeat those kind of results, they all use 2 doses. And when we speak to patients, they often tell us that they got more out of the second dose than the first dose. It's almost like the first dose acts as a prep for the second dose. So if the evidence is supporting in direction, why not do that? Our ultimate goal is to maximize the durability of effect. We're something here that has a benefit for months, not days or weeks. So that's the first thing. The second element is that for fast-acting antidepressants, FDA wants 3 weeks of data. So there's your primary endpoint at 3 weeks after the first dose. And then the last thing is just practicality. Just putting in integration sessions after the first dose, having your follow-up sessions, scheduling the patients for the next dose, just getting them through in and out of the clinic. You need that kind of time frame, 2-3 weeks, to make it all work for the clinic themselves. So all those things kind of came together. We're happy with it so far. It's running very smoothly. So the way things happen with the facilitators is, first of all, we have 3 different levels of screening for patients. So they're screened by the investigators to see if they're eligible to come into the study. And then they are screened again, blinded, by an arm's length third-party screener. It's using a tool developed by Maurizio Fava at Mass General, Harvard Medical School, that is designed to reduce placebo effect. Right? So important that we get the right patients. And then patients then, before they're randomized to either active or placebo, they have their first session with a facilitator or therapist. And that's sort of preparation, but it's also screening. What we've found here is that even though patients go through these sort of academic screening questionnaires, nothing beats a good hour-long sit-down with a facilitator to get to know the patient, right? And sometimes, not very often, but sometimes patients lie. And because people are trying to get into these studies to some extent. So that third screening, if you like, that first sit down, on rare occasions helps us catch folks that maybe shouldn't be in the study. Maybe there was a history of schizophrenia or something that didn't come up in the questionnaire. So it's a safety check. So after that, they then have 1 or 2 prep sessions with the facilitators. Some patients that have had long-term depression, 20, 30 years, need more than 1 prep session. It's hard to get them to sort of open up. It's hard to get them to talk about their depression. And you need to be able to set goals and intent with the patient. So sometimes it takes those 2 sessions. So that's done with a single facilitator therapist. During the dosing session, there's a second person in the room. FDA has asked us to do that. They're mandating that for all clinical studies for psychedelics at the moment. Our hope is that eventually when we come to market, and maybe even in phase 3, we can convince them that a single therapist or facilitator is enough, especially if it's a shorter duration and they're not there for 10 or 12 hours with LSD or 6 to 8 hours with psilocybin. Maybe you can use just one facilitator. And then there are integration sessions. The first integration session happens the next day. The next one's about a week after that. And we find that there's a gradual improvement in benefit from the first integration second to the second one. It takes a bit of time for patients to unpack everything that they experience in that dosing session. And the benefit seems to get better over time. So that's kind of how the study is working, and it's working very well.
Awesome. Just gonna go off on that. So with the two therapists, the two facilitators that you have currently, is it perhaps a psychiatrist and then maybe a licensed clinical social worker or a clinical psychologist? And also would love to hear your thoughts on the rationale behind letting patients remain on SSRIs or SNRIs.
Yeah, sure. So again, FDA has mandated who can be a facilitator in these studies. That's not up to us. So It has to be a qualified psychiatrist, psychologist, mental health social worker, someone with those sort of qualifications. The second person can be of a lesser level of qualifications, but still needs to have mental health or social work experience. And then, of course, all of them have been through our Embark training as well, to make sure that they're all kind of on the same page and level set. So we're getting the same behavior from all of the facilitators in the study and not variances across the study. Look, the SSRIs, SNRIs, it's just common sense, frankly. If we can show it's safe to administer psychedelics without taking people off SSRIs or SNRIs, why not? And there have been a few small studies that have not shown any toxicology or safety risks. We're not seeing any either. I think we have a safety review after each cohort. And there are probably more risks from titrating patients off of their SSRIs than leaving them on. Those rebound effects can last for up to a year. And also, let's not forget that some patients are getting placebo in these studies. So are we going to ask patients to come off their current treatment and then give them a placebo? I mean, ethically, that's challenging to do. So if we can develop protocols that are more convenient to prescribers, healthcare professionals, then I think that removes barriers to prescribing. And really, a lot of the thinking that's gone into developing CYB3 and CYB4, both the molecules, the formulations, and the protocols, are all about breaking down barriers and making these treatments scalable and accessible and reimbursable.
Awesome. Thank you, Doug. So the next question I have for you is, just would like to get your thoughts on the results that Small Pharma put out for their phase 2A in MDD with their, I believe it was intravenous DMT, and maybe some of the implications for Cybin CYB004 program in anxiety.
Sure, I think it's very helpful for us, that data. CYB4 is pharmacologically very similar to DMT. Obviously, it's deuterated DMT. It's pharmacokinetically very different to DMT. So how it gets in and out of the brain is different. What it does when it gets there is largely the same receptor binding profile. So that bodes well. Also, there's a big overlap between GAD, generalized anxiety disorder, that we're pursuing, and MDD. There's about a 60 to 80% overlap in symptoms and in diagnostic criteria between the two. Also, in reality, about 80% of patients with GAD also have depression. Wow. So as we go forward, as again the pivotal studies, I expect that we have the opportunity to look at two measures, anxiety and depression, with CYB4. And this, so I think the data from Small Pharma gives us a lot of confidence that we should see efficacy. But with our goal ultimately is to develop this different formulation, as you know.
Okay, awesome, thank you. And just coming back to CYB003, I watched you on the Dales Report. By the way, I really enjoyed that interview. It was very informative. And I noticed you were talking about CYB003 entering into a phase 3 trial sometime next year. I was just wondering, would it be possible to do the phase 1/2A and then proceed directly to a phase 3 next year, or would you also be doing a phase 2b trial?
No, we plan to get run to a phase 3. So the design study one part to really look at the study that 1, 3, 8, and 10 milligrams of CYB3. We had no psychedelic effects at 1 milligram. We had some psychedelic effects starting at 3 milligrams. At 8 milligrams, we're seeing quite robust psychedelic effects, about equivalent to 25 milligrams of psilocybin. So that gives you an idea of how much more potent CYB3 is. At 10 milligrams, we were getting 2/3 of subjects with 100% score on the visual analog scale. So the highest score you can get in terms of measuring psychedelic visual effects. We're now testing in patients 12 milligrams. And we may go higher. We'll see. We're assessing after each cohort, but each patient gets 2 doses. The first dose is either active or placebo. It's 12 patients in each cohort, so it's 9 to 3, and each patient gets a second dose is also active. So it means that if we dose all 3 cohorts, we'll have 63 exposures to active drug, and for a phase 2 study, that's quite a lot. And certainly enough safety data and efficacy data and dosing data to move into a phase 3, we think around about Q1 of next year.
That's awesome. That's really cool to hear. Thank you. All right, Adam, I'm going to pass it off to you.
All right. So to start things off here, so we talk about Cybin's financials for a second. So as of 12/31/2022, Cybin had about 23 million in cash. So you guys have also partially utilized 35 million at the market equity program, plus the recent 30 million stock purchase agreement with Lincoln Park Capital. Cybin seems to be burning about 10 million of cash per quarter. Is that a good run rate figure? And overall, how do you feel about Cybin's cash runway as we look over the next year or two?
Yeah, 10 million is about right. 10 million Canadian a quarter is about right. As we wrap up these current 2 studies, our external costs will go down, and our internal costs are fairly low. They're a fraction of that 10 million. But then, of course, as we move into next year, we'll be ramping up a phase 3 study for CYB3. We're looking at a phase 2 study for CYB4. We're looking at a PK subcutaneous study for CYB4. So as we ramp those up, we're likely to get back to that 10 million a quarter. So I think it's a reasonable number to use. And as we do much of this work externally with CROs, it's all discretionary spend, so we can time the spending of it. So it's pretty good assessment. So you're right; those numbers are right for end of December. I think a lot. We also shared some numbers mid-February where we had 20 million of cash still available. So in 6 weeks, we burned relatively little because of our ability to use the ATM. And we had 35 million Canadian left on the ATM at mid-February as well. So it was 55 million Canadian, and we have 10 million a quarter as a run rate. And we've just added another 41 million of capacity Canadian with the LPC agreement. So collectively, there's clearly about 2 years of capacity or so between those facilities. And we wanted with the LPC agreement to have a commitment. Look, nobody's happy with the share price is right now. Me especially. I'm a shareholder as well, of course, and heavily incentivized to drive the share price. So we wanted the commitment there, that 30 million US, 40 million Canadian, but we didn't necessarily want to access it all at once at these prices. And in the next several months, we'll have efficacy data coming from CYB3. We'll have dosing readout for CYB4. We're going to have news around the subcutaneous formulation for CYB4. Other regulatory news that I'm fully expecting from FDA as well. So a whole number of catalysts that we hope, along with the improving economic environment, will help drive the stock price up. And so we're not having to issue with all of these current prices. So great to have the commitment. That said, I think like everyone else, I'd like to see more directly on the balance sheet, of course. And maybe we can use some of these catalysts to drive that as well, in addition to these facilities that we have.
And Doug, sorry, Adam. What are the rules around Lincoln and their ability to sell these shares? How does that work exactly?
Yeah, look, they're a long-only fund. There are there are lots of less scrupulous funds that do these kinds of facilities. Lincoln Park is not one of those. They have a great reputation. They've done a lot of deals, 100 deals or so in biotech. They've done many repeated deals for companies, and companies don't go back to the same fund for repeat deals if they're not good deals. Right, so I think they've got a good track record and reputation. We did our due diligence. They're long only. They are committed not to sell short, and there's no warrants or other things associated with it. So I think it's a pretty good deal for us. It gives us flexibility, gives them flexibility, and at this point in time, being able to commit 40 million Canadian in the current environment, we think is a helpful move for the company.
Thank you.
Understood. Moving on, Cybin is developing CYB004, a deuterated version of DMT. Small pharma is also developing their own version of deuterated DMT. In this industry, intellectual property is everything. So in your view, is it possible for two companies to have unique patents on deuterated DMT and coexist side by side without infringing on each other's IP? Are these both considered different enough NCEs from one another where this won't be a problem? If you could share your thoughts.
Sure. I completely agree. IP is really important. We have almost 60 patents filed at this point in time across all of our programs. For CYB4, we have a composition of matter patent granted. It goes out to 2041. I can tell you that I am absolutely confident that these two molecules are distinctly different. And because we know pretty much what they're working on. I'm assuming they know what we're working on at this point in time. And I can tell you that during the preclinical process, the development process, we looked at a whole different range of deuterated versions of DMT. So the first is where you deuterate on the molecule makes a difference. So there's certainly locations on the molecule where deuteration doesn't impact the breakdown of DMT by monoamine oxidase at all. So it doesn't impact the pharmacokinetics. There are other locations where it does do that, and that's the goal, right? Is to improve bioavailability, improve brain penetration, but also slow the breakdown a little bit. Also, the level of substitution, so the number of hydrogen atoms that you substitute makes a difference as well. So we compared preclinically a whole range of different molecules, and we chose the one that we think gave us the greatest opportunity to move into a small volume injectable. That was the ultimate goal here. What we wanted to do is move away from being chained to an IV infusion pump. We think that infrastructure, the equipment, the staffing needed is part of what has created barriers to people accessing ketamine therapy, whether that's psychological or practical. Psychologists, psychiatrists just don't have IV infusion pumps in their offices. They don't use needles, right? They're not really those kind of doctors.
Yeah.
And so a small subcutaneous injection, a bit like a Wegovy or an Ozempic, which the patient can apparently give themselves, or the doctor could, or a nurse could very easily, unshackles the product completely from that infrastructure. That means that the CYB4 could be dosed anywhere that there's a bed or a sofa and a physician or facilitator there. And so, of course, we've not only got IP around the molecule, which is granted, and by the way, a range of other deuterated DMT and 5-MeO DMT molecules also in that patent. We also have IP filed around subcutaneous formulation and the specifics of that. And I can tell you that the subcutaneous formulation is a real challenge in that you're talking about a very, very small volume, 1.5, 2 milliliters, maybe less than that. And within that, there's only a certain amount of drug that can be loaded in there. So that drug better be potent. It better have good bioavailability. It better have good brain penetration. You're dealing with osmolality, pH, stability, small volume, and the matrix itself, the unique matrix that we put CYB4 into for release. There's all the complexities, but I'm pretty confident from the preclinical work that we're doing that we're generating good data, and we can have an interesting product here.
To build on what you just mentioned about the subcutaneous injections, what is your degree of confidence that a subcutaneous injection will be able to induce a full-blown breakthrough psychedelic DMT experience compared to the like IV administration and
Yeah,
doing it through
yeah,
IV?
that's a really good question. So psychedelic effects with psilocin, DMT, are threshold effects. In each case, and it's individual for each person, you've got to get them across a certain level plasma concentration threshold. It's not about ratio to body weight or anything like that. It's getting them across the line. So can we get them across the line with the subcutaneous? I'm very confident from the work that the team has done. Yes, we can do that. We're certainly seeing that preclinically. Then the next part is, where does the peak stop? You don't want it going too high that patients are in outer space. So you want to get them across the line enough, but not too far across the line. And then the third aspect is, well, how long does it then take to break down and get out of the body? And that's the final piece with the matrix that we put CYB4 into, is how we figured that out. And we're targeting that 45 to 60-minute kind of duration, so that's the last piece of the work that we're doing. So far, so good. It's drug development. It's still ongoing, but so far so good.
Understood. Ongoing debate in the psychedelic community is around the actual need for a psychedelic experience to achieve a therapeutic outcome. As I'm sure you're well aware, there are some companies trying to develop so-called psychedelic-inspired non-psychedelic drugs that increase neuroplasticity, etc., without the trip. In your opinion, how necessary is the actual trip, and do you think these new class of drugs without the trip will ultimately be successful?
Yeah, maybe at some point. Maybe at some point. The science never stands still. What we know today will be different than we know a year from now or 2 years from now. So at the moment, there's not a great deal of evidence to say that you can generate these neuroplastic effects without the psychedelic effects. Not seeing that, but that doesn't mean that that won't come at some point in the future. I mean, we don't even really know what the mechanism of action is of this neuroplasticity. We know that these molecules interact with a 5-HT2A receptor. We know that has downstream effects, and we know a lot of the mechanisms of internalizing receptors and all of the other effects it has downstream on neurons and other neurons. But we still don't really know what causes the neuroplasticity. So is neuroplasticity the same as the visual effects you have with psychedelics? We're making assumptions perhaps that they are, but we really don't know. So that's a question, I think, for another day, maybe even another decade. And for now, I think what we're focused on is what we do know, and focus on the clinical programs we have. And I think we've got 2 molecules based on well-characterized classic psychedelics. We've tried to tailor the modifications so that we're not messing with receptor binding profiles. We're simply altering the pharmacokinetics to improve the patient experience, improve the practical use of them for physicians, make them more reimbursable. And that's a hard enough task as it is. So I'll leave this other theory to the scientists.
Understood. Moving on, there are a lot of questions around the commercialization of psychedelic assisted psychotherapy. Drug pricing is also notoriously non-transparent. Let's say, for example, CYB003 gains FDA approval and becomes insurance reimbursable. How will the economics be shared between the drug company and the clinics administering the therapy? Assuming the total treatment costs, say, $20,000, how much would go to Cybin and how much would go to the clinic for providing the service?
Well, first of all, I think it's impossible at the moment to say what pricing will be. It'll heavily be dependent upon the profile of the drug, especially in the US. Outside the US, with single payer markets, it's going to be a little different. But in the US, if we can deliver 6 months of remission for patients, that's a very different value proposition than a week or a month, as we might see with say ketamine. Patients that are in remission for 6 months are less likely to be in the hospital, less likely to be taking other drugs. Other comorbidities are likely to be improved. So there's a big case for large pharmacoeconomic gains from durable results, but we just don't know yet, right? What these what these effects will pan out at. Will it be 3 months, 6 months, 12 months? We really don't know. So I don't mean to skip the question there on pricing, but that's just the reality at the moment. It all comes down to what payers will pay, and obviously the greater value, the more they pay. Look, when it comes down to commercialization, we're not planning to own clinics or operate clinics. We're planning to supply the drug, and then healthcare providers will deal with the administration. Our goal from the outset is to try to make that administration as low cost as possible. So half the time in the clinic, say, with CYB3 might be half the clinic costs. If we can reduce to one facilitator versus two because of that, that's half the staffing costs. If we can move to a subcutaneous formulation instead of an IV infusion pump and equipment and staffing, that's reducing costs. And if we can deliver durable effects over long periods of time, that's obviously very economically beneficial. So, I mean, although we don't control the protocols, we're trying to develop into our studies protocols that make sense, commercially. So yeah, I mean, I think we'll be somewhat removed from the administration, but obviously helping with guiding how these drugs should be administered.
And,
Understood.
Sorry, Adam.
And also, I guess just to color this a little bit more, so do you think can you think of any similar arrangement in existence today in our medical system? For example, like cancer immunotherapy treatment, would that be a good example in terms of maybe like a model that it would be kind of similar to?
I think that there are some examples of drug companies or device companies integrating with delivery of care. Like in dialysis, Baxter and Fresenius have their own dialysis centers. Dermatology. In dermatology, the drug is supplied, the treatment is supplied from the drug company, and then the dermatologist gets reimbursed for the drug, but also, the administration. It might be a procedure, or it might be an aesthetic procedure, it might be a removal of something, it might be an implantation of a device. So these things have always somewhat been separated. Our goal is to make sure we can create protocols that are practical for physicians. They need to get reimbursed for their time, obviously.
And Doug, I'd like to ask you a little bit about the clinic space here. I've heard, for example, Florian Brand, multiple times he stated that he sees these psychedelic therapies mostly being administered within the existing infrastructure, with set out by J&J for esketamine and ketamine. Do you share his same views, or do you think these therapies will mostly be for psychedelic focused clinics only?
I think it'll be a mix of new clinics and a mix of existing. Providers are always looking for ways to expand their business. I know plenty of primary care docs that shifted their primary care business to Botox when Botox came along, because it was profitable for them. And I think you'll have centers like TMS centers, for example. There are hundreds of those across the US where they're regularly seeing depressed patients looking for alternative treatments to SSRIs. They'd be the ideal type of patients to perhaps consider psychedelics. So if we keep the barriers down with these protocols and with the administration, I think there are a lot of centers that could adapt. I mean, at the end of the day, you just need, as we have in our clinical studies, a comfortable room and a bathroom. These aren't complicated spaces. Now you start getting into IV ketamine or maybe IV DMT, where the hookup is very different. Then that's going to limit administration sites because of that need for equipment and training, insurance, liability, all those other things that come along with that. But long answer, but I think there'll be a bit of mix of both. We've seen plenty of new ketamine clinics spring up. I think you'll see dedicated psychedelics clinics, but you might just see others, therapists and psychiatrists, psychologists, figuring out that they can use these tools to enhance their care for patients instead of just giving talk therapy or psychotherapy. Perhaps then they can accelerate that therapy by bringing psychedelics into their practice.
Yeah, and speaking about bringing it into their own practice, I'm aware of a company called Journey Clinical who is doing some work to make it so. I think they're already doing it with ketamine lozenges to enable practitioners in private practices to administer these type of therapies. Do you see that happening right away, or when these drugs are approved, or do you think that's more a little bit later on when, let's say someone with their own practice can have someone take these psychedelics under their supervision?
Yeah, no, I think if someone's trained and their facility is adequately licensed, then no reason that these treatments couldn't be given pretty much anywhere. I mean, like I said, the rooms that we're using for our phase 2 study for CYB3 are not complicated. You know, they're very comfortable. There's a sofa, a bed, there's cushions on the floor, there's nice lighting and music. You know, they're very comfortable and relaxed. As a bathroom. Down the hall, there's a crash cart, of course, right? Because it's a medical center, but the rooms themselves and the facilities needed are not complicated, especially for a CYB3, which is oral.
See.
So I just wanted to touch on CYB004 again for a second. So you mentioned not wanting to send the patients into outer space. So where exactly are you trying to get them in regards to the DMT experience for the work you guys are doing?
Yeah, so anything over about a 7 on the visual analog scale appears to correlate with 1, a robust psychedelic experience, and 2, about 70% receptor occupancy for these 5-HT2A receptors. And that's true for psilocin and DMT, which are chemically very, very, very similar. So getting patients above 7 and as many patients above 7 as possible on that scale is the goal with CYB4 and with CYB3. You know, there's always going to be some patients that don't respond. There's always going to be stragglers with any treatment in any clinical study. My goal is to maximize how many patients we can get across that threshold without hitting the upper limit of side effects. And with DMT, if you go a little too high, people get anxious. You know, it becomes a non-pleasant experience. So there's always an upper threshold. And that's been the goal of our phase 1 study so far. We've dosed, I think, 64 exposures of DMT so far. So we have a pretty good idea of where the bottom and the top thresholds are. And then we're correlating that to CYB4.
Thank you. And also just one more thing in regards to the patients who will be remaining on SSRIs, SNRIs, are you finding that you are having to dose them higher due to their serotonin receptors being occupied by the antidepressants that they're on?
That is a really good question. You could assume that we thought that might happen, which is why we went from 10 milligrams in our phase 1 to 12 milligram dosing in the phase 2 in patients. We were trying to see, well, maybe they might see a little bit of blunting effect because of the SSRIs, SNRIs. I can't say that we're seeing that. Now, the study's all blinded, right? So it's a little hard to say, but we're seeing very robust psychedelic effects. In what portion of patients, again, blinded, we don't know. We may even push the dose a little higher to see where the upper end is. It's always good to have a good dose ranging. You know, I feel like when I'm looking at some of the academic studies and some of the other studies on psilocybin, that people kind of landed on 25 milligrams. I haven't seen really robust dose titration. It might be that 26 or 27 or 28 is better for most people to get them across that line. I haven't really seen those studies done. So one of the benefits of having a new molecule, a modified molecule like CYB3, is we've had to go through those steps. So we're on our 5th dose level. We might go to a 6th, and we'll have a really good curve then to try and understand what the best dosing might be.
Yeah. Speaking of 25 milligrams, Doug, are you aware of, in the Compass trial, what % of people actually reached the psychedelic state? I was talking to my friend about this, and he's convinced that there might have been a lot of people who really didn't get there with that dose. Do you know any data around that by any chance?
Yeah, I haven't seen that. I think we've only seen fairly top line data, but that wouldn't surprise me with psilocybin. It's fairly variable, and that's been widely known for quite some time. So that would be another reason to push the dose up potentially, or a reason to have 2 doses or 3 doses. I think there's several ways to address it. We've tried to address it through changing the molecule in the way, and so it's more bioavailable and has better brain penetration, getting more in the brain. We are likely able to push the dose a little higher. because we're able to test this dose curve, and of course using 2 doses. So all these are little tricks, if you like, in the protocol to try to make sure as many patients get over that line as possible.
Awesome.
Awesome. Well, that's all the questions I have for today. I think Jake is also finished as well. Before we wrap things up, if you would like to address the community real quick, I'll give you you have the floor right now, Doug.
That's great. Well, thank you guys for all the questions. It's been a long time, so it's great to come back on and have a conversation. Look, it's been a tough year, I think, for the whole sector, and for shareholders and for our team who are working hard every day. It can be pretty demoralizing when the share price is down. I get that. You know, we are working, executing every day. I think we've hit every milestone that we've said we would hit on time, and we plan to do that going forward continuously. And we have, I think, some pretty interesting milestones and value drivers coming up over the next 6 months. Between now and the end of the year, you know, we'll be in a position where we hope to have completed phase 2 for CYB003 with efficacy data and have a plan in front of the FDA for phase 3, ready to start that in Q1. When you consider we went from the bench to the clinic in 18 months and now pretty aggressively moving through this phase 2 study with great recruiting into a phase 3 within a little over a year. This is moving actually pretty fast. It may seem slow to folks on the outside, but because drug development is slow, but this is actually moving very, very fast. And as for CYB4, I'm bound to say this, but I think it has the potential to be the market leader. It has an onset of action in 2 minutes. It has a peak at the moment we're seeing of about 12 minutes to peak effect. We'll likely end up with a designed duration of 45 minutes or so in a simple subcutaneous, non-invasive formulation that can be given anywhere. And I don't think there's anything else out there right now that matches that profile. So we're pretty excited about that, and you know, then the next steps for that, which will be a phase 2 study early next year.
Awesome. Amazing to hear. So I want to thank everyone for watching this interview. Really appreciate you guys. I want to thank Doug for coming. And I also want to thank Jake for being a part of this with me. So I hope you guys all have a wonderful day. Like and subscribe if you enjoyed this. And yeah, have a great day, guys.
See you guys. Thank you.